So we're going to talk about describing data, distributions, and variance. These are really fundamental concepts. You want to get a really strong grasp of these to go forward from here, okay? Describing data. How do we describe our data? We got some data. We want to describe it. When we start off with data, we have a whole bunch of numbers. Maybe we have a whole bunch of numbers in a spreadsheet or on a piece of paper, but it's like a long line of numbers. And our brains really can't handle that much complexity. So we need to figure out how to reduce the complexity to summarize the data into a few key numbers so that we can understand it. So just a big overview, we're going to talk about the key differences between populations versus samples, properties of distributions. And if you're not clear on what I mean by distributions right now, you probably will be by the end, okay? Means and standard deviations, two really fundamental, essential statistics for describing data. And then properties of the normal distribution, specifically the 68, 95, 99.7 rule, and why being normal matters. Then we're going to go over histograms, box plots, and quantile plots. And after the lecture, we're all going to work on doing a histogram in class, a histogram and a box plot in class by hand. I know we already did this with R, and it's really easy to do this on a computer, but I want you to do it by hand at least once, maybe more than once, so that you can really get clear in your mind about what this is and what this graph is showing, because we're going to use it constantly throughout class. So a population is the entire group of individuals. It's measured with a census, like the US Census. They measure everybody. They try to count absolutely everybody. They go to every single person every 10 years and they try to count them. They only do it every 10 years because it's incredibly difficult and incredibly expensive. Very rarely do people try to conduct a census of an entire population. It's more likely that people take samples. So the population mean, here's some fun Greek symbols. Who here speaks Greek? No, not me. No, it's Greek. I don't know Greek, but there are a few symbols we have to know in Greek. They'll come up again and again, so just put them in your brain right now as much as you can. Take notes. The population mean, and actually since we don't use populations that often, we don't see this that often, but every once in a while you will, is the Greek symbol mu. Looks like a little U. That's mu. And the population standard deviation, and we'll get more into what a standard deviation is the Greek sigma, that symbol sigma. And that's like the S, right? So the mu is like the M and the sigma is like the S. And that helps us to remember that when we're talking about a sample, now we go out of Greek. And when we talk about a sample mean, we use this symbol X with a bar over it, and that's just called X bar. X bar. If you're trying to type in Word or another software and you're like, how do you make this symbol? You can just Google how to make X bar. It'll come up. That's what it's called. And the sample standard deviation is a simple S, just an S. Easy, right? Again, we hardly ever measure populations. If the example I usually think of when I think of measuring a population is the alala, the Hawaiian crow. It's an extremely endangered species. It's so rare, they're all in captivity. So theoretically, you could measure all of them. They're all in captivity, right? If you wanted to know the weight of all the alala, you could measure all of them. But let's say you wanted to measure something more common, like the apapani, a very common Hawaiian bird. You can't measure all of them. They're flying around all the time. And then by the time you start getting halfway through, some are going to die and other ones are going to be born, right? It's impossible. So you take a subsample of the entire population. If you've done your sampling properly, your sample should represent the population pretty well. Okay, distributions. There are three fundamental properties of distributions that we will look at in class all the time. One is the shape, two is the center, and three is the variability. So here's an example of a bunch of different distributions that you can see the different shapes. And the shape is really important because the shape tells us what kind of statistics might be appropriate for this data. So if you're not sure about what the word distribution means, I think of it like this, like distributing something, right? Let's say you're distributing cards to people around a table before you play cards. One card for you, one card for you, one card for you, right? You're handing them out, you're distributing them. And let's say one person gets more cards, that person's in the middle, they get the higher stack. People on the edges get fewer cards, they have the lower stacks. And if you were to draw that like this, you would have this symmetrical distribution. Now, this particular distribution shape, how the data are distributed, how many points are right around the mean of 4 and how many are close to the minimum and close to the maximum, is important for trying to understand properties. And we'll get more into that about when we talk about properties of normal distribution. But for now, just remember that when we're looking for normal data, we're looking for this symmetrical, unimodal, bell-shaped curve. Symmetrical, both sides are more or less the same. If you cut it in half and fold it, they would sort of match up. Unimodal, there's just one mode, there's just one peak, right? And it's bell-shaped. It's kind of got this short on the edge and tall in the middle, roughly bell-shaped. That's what we're looking for when we're looking for a normal distribution. And we'll go more into that this week. There's other kinds of distributions. You'll see these too. They're very common in nature. There's the skewed right distribution, where you have a lot of data that are very small and a few that are very large. There's also skewed left, when you have just very few small data points, or data points with small values, and you have a lot of data points with large values. These might just be the nature of the data that you're collecting. There's also non-symmetrical bimodal data over here on the right. So you've got 2 humps. That's bimodal data, 2 modes, right? 2 really common values. You can't analyze them using the same statistics because they don't follow the same assumptions, the same properties. And there's also symmetric bimodal data. You could have it be very symmetric, but still be bimodal. And then there's uniform data. So you've got all the data points have the same distribution. You have just as many 1s as 2s as 3s as 4s as 5, etc. That's pretty uncommon, but it could happen. So different data inherently have these distributions. If you're wondering, well, why do we want to look at normal distribution? Well, the normal distribution is just very common in nature. It's just very common. It's found in so many places that it became the sort of basic fundamental assumed distribution when people were looking at data. They're like, this is happening all the time. This is how we see data just show up. When we take data on, for example, human height, it just happens to be normally distributed. You have very few people who are very short, very few people who are very tall, and most people are somewhere in the middle. Just how it is. But some types of data are, by their nature, skewed. For example, if you took a bunch of guinea pigs and you inoculated with a pathogen and you wanted to see how long they survived, that's so mean, but this is the example from the book. Anyway, it would probably be right skewed. The survival time of most guinea pigs would be short, and a few would last longer. It's not wrong. There's nothing wrong with these data. It doesn't mean that they're bad because they're not normal. It really just tells us something about the data and about the study that we're doing. Okay, central tendency. There are 3 measures of central tendency that we'll use a lot. I wonder if I can minimize this, get that out of the way. Okay, there we go. 3 measures of central tendency. For normally distributed data, that nice symmetric unimodal bell-shaped curve, you want to normally use the mean. Everybody here knows what a mean is, right? We're normally talking about their arithmetic mean. There's other kinds of means, but the mean that we normally use is you just add up all the values and you divide by the number of data points, right? Pretty simple. For skewed data, For data that's not normal, it's usually more appropriate to use these other two measures of central tendency, the median and the mode. These are also really, really easy. They're actually easier than the mean to find because you don't even have to do math. For the median, you just put all the data in order from smallest to largest and you find the middle point. If the middle point is 2 data points, if the number of data points is even, you just average those 2 data points in the middle. And the mode is just the most commonly occurring data point in a data set. And that can be really handy. And so, in fact, sometimes the mode is what you want. Like you want to know, like, let's say you want to open an ice cream stand and you want to find out what's everybody's favorite flavor of ice cream so you can be sure to stock that a lot. And so you go out and you do a survey and you ask people, what's your favorite flavor of ice cream? And you want to know what's the most commonly occurring value. Do more people say chocolate? Do more people say vanilla? Do more people say Rocky Road? Right? In this case, the mode, ice cream a la mode, is the most interesting summary statistic. Even if you had normally distributed data, that's what you'd want to know. And I don't know why these numbers all come out as 1. It should say 1, 2, 3. So funny when you change it. Like I have the PowerPoint and then I load it up to a different software and it changes. Okay, measures of variability. So measures of central tendency shows you sort of where the center of the data is. Measure of variability helps you to see so the spread of the data. There are, oh my gosh, these numbers are all mixed up on here too. Sorry about that. I'll fix this when I upload this for you for La Lima. But so the first measure of variability that is really commonly used for both normally distributed data and non-normally distributed data is the range. Just a maximum value minus the minimum value. The variance, and here's how you do the variance, and we're going to do this by hand on Thursday. You just, step 1, you find the mean. Step 2, subtract each value from the mean to find the deviation from the mean. Then square each deviation, add up all the squared deviations. That's called the sum of squares. And then you divide by those squared deviations. You divide the squared deviations by the number of data values minus 1, n minus 1. That's called the degrees of freedom. So pretty simple, right? You can do that with a calculator. And if you take the square root of the variance, you have standard deviation. And yeah, it's interesting on my slide, my square root symbol didn't show up. And then if you want to take the standard error, yeah, this didn't show up right on my slide, but it should be the standard deviation divided by the square root of n. I'll fix this when I upload it for you, maybe because I went back and forth between Google Sheets or Google Slides and PowerPoint. And the coefficient of variation, which I really like. I like the coefficient of variation. I don't see it used that often, but I think it's handy because it helps you to scale or standardize amongst lots of different data sets. It's the standard deviation divided by the mean. So you might have a data set that has a really high standard deviation and a really high mean, and another data set that has really low standard deviation and really low mean. And it's kind of hard to compare them. Which one is more variable, right? Well, if you divide the standard deviation by the mean, it helps to give you an idea as a percent how much variation there is in the data. And then you can compare them. Okay, measures of variability. This particularly works for skewed data, but we also use it for normal data. It's something called the 5-number summary. We're going to use it a lot. 5-number summary. What are those 5 numbers? Right here. Minimum. Straightforward. Smallest number. We know this. 2, the first quartile. That's the median of the first half of the data. A little bit more vague, but we'll get that. The second quartile, or Q2, is the median. Again, the central data point. Line them up from small to big. Take that middle one. The third quartile is the median of the second half of the data. Well, now that we understand what median is, that's starting to make more sense, because if we think about it, if we've lined them all up from the smallest to the largest, and we've taken the middle one as the median, and then we take that smallest half of the data and we find the middle one of that, that's our first quartile. And then we take the bigger half of the data and we find the middle one there, that's our third quartile, our Q2. or Q3, right? Q2 is median, Q3 is the middle of the bigger half, Q1 is the middle of the smaller half. And max, you guys know what maximum is. You've known that since you were kids. Now, the interquartile range, IQR, is just a fancy way of saying take Q3 and subtract Q1 from it. It's the difference between that bottom part of the box and the top part of the box. So if the Q1 was 5 and the Q3 was 8, then your IQR would be 3, right? Straightforward. Now, outliers. Outliers are typically defined by a very specific rule. That is the 1.5 times IQR rule. So simply stated, any observations that you see in your data that are more than 1.5 times that interquartile range, Q3 minus Q1, right? Below Q1 or more than 1.5 times IQR above Q3 are considered outliers. And this is where it can be a little bit funny when you're looking at a box plot, because sometimes you see these box and whisker plots and there'll be a dot up here or a dot down here that are showing outliers. But it's like, wait a minute, if that's the maximum, how's there an outlier up here? If that's the minimum, then how's there a number that's less than the minimum? That doesn't make sense. That's not right. Okay, you're right. That doesn't make sense entirely. So typically when we're making the box plots, the minimums and the maximums are actually defined by this 1.5 IQR rule. And that's where you put your minimum and your maximum. And if there's anything smaller or larger than that, then you put a dot. Does that make sense? Okay, we'll do this more. Okay, I just wanted to show you again how you find Q1 and Q3. So again, Q1 is the data point exactly halfway through the lower half of the data set. You find it by finding the median of the data below the median. Q3 is the halfway point from the median to the end of the data set, and you find it by finding the median of the half of the data above the median. So again, if you've got your median here, you look, what's the point halfway between this median and the minimum? And that's Q1, and the point halfway between the median and the maximum is your Q3. And then if you have outliers, it would be, so yeah, I like this little graph. This particular one nicely shows you that minimum in quotes is actually Q1 minus 1.5, the interquartile range. Anything smaller than that is an outlier. Maximum might not be the biggest number. It's actually Q3 plus 1.5, the interquartile range. And there might be bigger numbers than that. Okay, so minimum and maximum are not necessarily the smallest and largest numbers. They're relative to the interquartile range. All right, so again, we're talking about the 5-number summary. So if you look at this box plot and you put it onto that histogram, that bell-shaped curve, you can see that it's defining a lot of properties of the data that we might find interesting. For example, that interquartile range, that distance between Q1 and Q3, contains 50% of the data, right? Half the data is right there. Boom. Now, that might be of interest to you if you just want to find out where most of the data points lie, right? And again, if the data are not normally distributed, that won't be true. Imagine drawing a skewed curve onto this slide, onto this picture. It would no longer contain 50% of the data between the Q1 and Q3. It only works for normal curves. And that whisker, that Q1 minus 1.5 times IQR is going to contain another almost 25% of the data. And that next whisker is going to contain almost another 25% of the data. So the vast majority of the data is contained right in there, which makes sense. It should be, with maybe an outlier or two outside of that. Not only that, but you can see that typically we've got the standard deviation of the population falls pretty close to that interquartile range. If we go 1 standard deviation from the median and 1 standard deviation below the median, pretty close. Okay, accuracy versus precision. These words are often used interchangeably, and I'll be honest, I sometimes get them mixed up myself. I'm like, wait, what's the difference again? And this is what kind of helps me to remember. Accuracy hits the bullseye, precision hits the same spot repeatedly. So why are we talking about this besides just an interesting vocabulary lesson to help you with your GREs if you're going to grad school? It's because the mean relates to accuracy because it describes the center of the distribution, right? It hits the bullseye, the mean. The standard deviation, S, relates to precision because it describes the spread of the distribution. How far apart are the different data points, right? So you can be accurate and precise. You have all your points tightly clustered in the center. You can be not accurate and precise. All your data points fall in the same place, but they're way off. You can be accurate but not precise. You're always around the center but never that close to it. Or you can be not accurate and not precise where your data points are all over the place and not anywhere near the center. So these are useful tools for comparing samples and populations. The means and the standard deviation, if you have a normal curve and you have the mean and the standard deviation of it, you can describe the entire curve. You know what it looks like, right? You can draw it from there, assuming it's normal. If it's not, it's all bets are off. I think we looked at this one in the last lecture too. So in this graph here on the right, the means are all the same. You see the peaks are all in the center, are all right here. They're all lined up. The means are the same. There are 15, but the standard deviations are different. So a population distribution with a smaller standard deviation will have a smaller spread, a smaller range. And if it has a larger standard deviation, like this red one on the outside, it has a wider range, which means it's going to have a flatter distribution as well. So these different shapes tell you a lot about the data. Just by glancing at them, you can kind of see something about them. Like this is a less variable population. This is a more variable population. Now, if your means are different and your standard deviations are the same, so you can see here, the width of the bottom of the bells are all the same for these three population distributions, but the means are different. So the centers are in different places. For normally distributed populations, the mean will fall right in the center, okay? If it's skewed, it'll fall somewhat off. Okay, the 68, 95, 99.7 rule. Okay, all normal curves share these same properties. And we don't have to do the math of integral calculus. We can just see that it's there. So if you have a population with a mean of 0, and you go 1 standard deviation below it and 1 standard deviation above it, you will get 68% of the data. If you go 2 standard deviations above and below it, you'll have 95% of the data. And if you go 3 standard deviations above and below it, you'll have 99.7% of the data. 99.7%, quite a lot, right? So if we really pay attention here to this middle one, this -2 standard deviations to 2 standard deviations, 95%. So 100 minus 95 is 5%, right? So only 5% of the data lie outside in these tails. Most of it is contained here. So imagine you were looking at a population of students and you would say most of the students, their heights are right in here. Very few students have heights much taller than that or much shorter than that, right? So if you were going to choose a student at random, most of the time, 95% of the time, your student will have a height in this big part of the bell-shaped curve. 95% of the students' heights will fall between 2 standard deviations of the means, right? So you're most likely to randomly pick a student with a height in this range. And you're very unlikely, only 5% of the time, are you likely to randomly pick a student with a height larger or smaller, right? Why is that important? Because that relates to p-values, which we'll be talking about more next week. Now, again, if the data are not normally distributed, this rule does not apply. If the data are skewed, then a different amount of data fall within 1 or 2 standard deviations. And so we have to be very careful when we're analyzing data based on the assumption of normality that it really is normal, at least approximately normal. So how do you know if near normal? This is such a hard question. What is normal? Am I normal? Am I not normal? So one way to assess your data normality is to plot the data on a normal quantile plot. And we don't have to calculate this. We're going to have the software do it for us, but we need to know what it means. So what this does is it takes each data point in our data set and it ranks it. And the percentile ranks are converted to z-scores. I'll talk about z-scores on the next slide. Then the z-scores are plotted on the x or horizontal axis, and the actual values are plotted on the y-axis. If the data have an approximately normal distribution, the normal quantile plot will have a roughly straight line pattern. Okay, so just a little bit more about what a z-score is. A z-score is a way to standardize the variation to compare populations. So a z is just the data value minus the mean. And in this case, we're using mu, population mean, even though in practice, it's probably going to be x bar, the sample mean, divided by sigma, although in practice, again, it would be s. It would be the sample standard deviation. So it's just a scale. It's like, what's the difference between the data point and the mean divided by the standard deviation? If x is 1 standard deviation larger than the mean, then z equals 1. If x is 2 standard deviations larger than the mean, z equals 2. So when x is larger to the mean, z is positive. When x is smaller than the mean, z is negative, right? And if we look at that here on a plot, we can see, again, our z-score on the left-hand side is plotted on the x-axis, and actual values of rainwater pH value on the y-axis. And this is approximately normal. If it was perfectly normal, it'd be a perfectly straight line, but more or less the data points fall in a straight line, the z-scores versus the p-values. Whereas here, when we have a very right-skewed data, our z-scores versus survival time in days, you can see it's a curve. That's not a straight line. It doesn't match. It would be a non-normally distributed data. There are also normality tests that we can use, and we'll go over these next week. One thing important to recognize about normality tests is that the null hypothesis, and I know we haven't talked about that yet. I'm introducing this idea now of null hypothesis, which is your hypothesis that nothing's happening. Their null hypothesis is that the data are normally distributed, and that your alternative hypothesis is that the data are not normally distributed. So if your p-value is small and you accept your null hypothesis, if your p-value is large, that's good because it means your data are normally distributed. So the normality test asks whether the data you sampled are consistent with the assumption that your data were sampled from a population with a normal, or otherwise known as a Gaussian distribution. So we'll look at the graphs, the probability plots, the histograms, and not just the results of the normality test. We'll take all of these things into account when deciding about whether or not our data meet the assumption of normality. Okay, that is the lecture. Now what I want to do is for us to get out our pencils and paper and make histograms with our data.